what you should do or should have done in the practical for those measurements is label the leaves, because otherwise the data will really get messed up. So what I usually asked, or what the groups really usually did, was labeling the leaves starting from the lower side, A, B, C, D, E, and so on. And then the individual plants, they also have names. So it's either mycorrhiza, control, mycorrhiza in drought, or drought without mycorrhiza. And once we've done that, so the leaves, they have names. You don't need to write them on the leaves, but you just need to make sure, discuss with your fellow students that you know which leaf you're working on. And why that's important is, for example, I'll show that in the next clip. One thing you need is the leaf area, and so this will be done usually by other people, and you need to link the porometer measurements to a specific leaf. And so you know that values from this leaf, for example, which would be A, B, C, D, leaf E, I think, the fifth leaf starting from below, and this has certain porometer values. These should be linked to the leaf area, which I explain in another movie, and only then you can compare the measurements for the whole plant in the end to the scale measurements. By the way, now I'm talking about measurements for the whole plant because I asked to compare transpiration among the different treatments, but based on the size alone, you would know that transpiration will be much larger for this one, for example, than for that one, right? Because the plant has just a much larger leaf area than this one in total. So of course it will lose more water, normally. But actually, so that's one of the questions you can answer, or you should answer in the practical, in the course notes, I mean in the report. But what's maybe more interesting, and you should definitely do as well, is calculate the transpiration in total of the whole plant per unit leaf area. So that means that once you have the total transpiration rate for the whole plant, you should divide it again by the leaf area. And then it's interesting, because then maybe it's the other way around. Maybe the one of this one will be higher than the one of that one. And of course now I'm comparing two single ones, but in the end, normally I think we had 3 replicates. Here I just took 2 upstairs. And you can compare them statistically. Okay. Having said that, we can start doing some measurements, and I can show you basic principles of a porometer. So that's how you turn it on. Nothing special here, I think. And here, well, with this we will do the measurements. You see on the right here, so on the metal side, there is a hole in it. And that's actually a chamber in which measurements will occur. So the other side is just for holding something. So later we will put this over a leaf. And for example, in this case, the metal, you should always remember the metal piece, metal part, is the part that is measurement. So in this case, we're measuring the lower side. Maybe it's not that clear, but it was the lower side of the leaf. All the way around, the upper side. Okay. So you want to know both sides. Depending on the plant species and where in the plant we are, you can have transpiration and stomatal conductance on both sides of a leaf. Okay. Now before going to the actual plant, first just explain a bit more principles about the device. So you have this chamber, and of course when a chamber is attached on a leaf, what will happen is, so like this, well, what will happen of course is that first the chamber is in the open air, and the water concentration, water vapor concentration or pressure will be the same as in the outside environment, so it's in equilibrium with that. But when I put it on the leaf, what will happen is normally, so the leaf is more wet, and when stomata or some of the stomata are open to some extent, water will evaporate, transpire from the leaf to the chamber. And what this device does, it detects that at a certain point there's a threshold, and what it will do is just count 2 minutes. And then based on how much this concentration of water vapor has increased in the chamber, so it has a sensor there, it knows what the stomatal conductance is. Or actually the transpiration, but this device was calibrated earlier. I did it earlier in this room, and so it gives an idea of the stomatal conductance. But so the principle is just, you put it on a leaf, and you will see, I think it's 2 minutes. You need to wait for 2 minutes. Water is diffusing into the chamber, and then in the end, it gives an idea. It's just you get, internally it's just seeing a slope, and then it calculates a rate which is translated to a stomatal conductance. All right. Now in your course notes you have a question, and one of the first ones is, I think, to measure a dry filter pad. Over here, these are filter pads. It's just nothing more or less than a piece of paper. And it's to measure a dry filter pad. So that means I will put the device around that. So now you don't see me doing that because I have the camera in my other hand, but this is what I did. And now, I need to press enter. And it says, place sensor head on leaf, but actually nothing is happening. Why? Well, of course, because this is in equilibrium with the outside environment. I mean, there is no water going from this to the air around it, or the other way around, because it has been there all the time. So it's in equilibrium, so you will measure nothing. There is no net transport of water from the piece of paper to the porometer, to the chamber of the porometer. But now, maybe more interesting, there's another question to do the same, but do that with a wet filter pad. Over here, this is some distilled water. So what I can do is I can just wetten this. I need to place the camera down for a moment, but I'm still there. Sorry. And then, there we go. So now I added some distilled water to this. And again, try to measure this. Now what should happen normally, you see, so now it passed the threshold, and there will be water coming in, water vapor. And now sorry, I told it was 2 minutes, but actually it's just 30 seconds. There is a measurement, and you get something in millimoles per square meter per second. Well, that's the measure of the stomatal conductance. Yeah, exactly. So it gives, if this were a leaf, its stomatal conductance would be 777 millimoles per square meter per second. Now, of course, this is not a leaf, but still there is some resistance. I mean, what I mean is there's water on there, but it does not like evaporate in a millisecond. I mean, not infinitely quickly. There are still some things that are blocking the water here or preventing the water here from escaping in no time, so to say. And that is, of course, the thing I already discussed. That's a boundary layer. Well, it also depends, of course, on the other environmental conditions, so the temperature here, and in the end, the water vapor gradient between the surface, which has a relative humidity, again, of 100%, and the environment, which has a lower relative humidity. But here in this room, the same temperature, because our leaf is not in the sun. In the outside environment, by the way, of course, leaves, they're often in the sun, and their temperature will be higher often than, but not necessarily, can also be lower, but can be higher or lower than that of the outside environment. And then you need to take that into account into the calculations. But in this case, in our room here, it's the same. But so what this now actually gives us is a boundary layer conductance. Because there are no stomata, but the only thing limiting the transpiration, or the evaporation in this case, is the boundary layer. Of course, in that device there is no wind. So that's why if there is wind, this thing is, of course, is not correct. And then you have a formula for that. But in this case, you will have a value. Now you don't need to use this value for your calculations. Please use the one you had from your colleagues who did the practical earlier on. Or if there was wind during their practical, you use the formula which I gave, along with the wind speed, which they should normally have given you or should give you.